Hi, I'm Ashley Reid, and today I'm going to be talking about how we can use ancient viruses to kill cancer as part of the Evidentic Scholarship of 2021. Cancer is a widely pervasive disease, with individuals in the United States having a roughly 40% chance of developing cancer over the course of their lifetime. This equates to between 1 in every 2 or 3 people being diagnosed with cancer at some point. So given that this is such a massive challenge, how do we begin to treat this disease? There have been 10 different properties that have been identified as key for cancer cells to survive and thrive, and these have been called the hallmarks of cancer. Targeting any one of these hallmarks of cancer has the potential to be used as a treatment for patients. One of these hallmarks of cancer is the ability of cancer cells to avoid immune detection and killing, and cancer immunotherapy aims to target this by making cancer cells more targetable by the immune system. Cancer immunotherapy has become a frontline treatment for tumors such as melanoma and lymphoma. And here I'm going to discuss a potential new combination immunotherapy using engineered CAR T cells and epigenetic drugs in combination to overcome weaknesses that each of these therapies have by themselves. T cells within the immune system have the primary function of killing infected or cancerous cells, and antibodies in circulation are known for their specificity. Antibodies have a very low dissociation rate, which means that an antibody is roughly 1 billion times more likely to bind to a target than it is to dissociate from that target. CAR T cells are engineered to combine the specificity of antibodies with the killing ability of T cells and direct that combination against cancer cells. Kymriah was the first FDA-approved CAR T cell therapy in 2017. It's used in lymphoma and targets CD19 expressing cells, which means that it kills all B cells in the body, both cancerous and healthy cells. This was a super successful immunotherapy. There was a 64% of patients showing a response, and then 60% maintained that response up to 5 years following the treatment. Because of this initial success with Kymriah, 4 additional CAR T cell therapies have then been approved by the FDA. CAR T cells are amazing in lymphoma because there's this unifying target. But there are other challenges with moving to other tumor types. First, solid tumors often have an immunosuppressive microenvironment, and that can exhaust CAR T cells and prevent their ability to successfully kill tumor cells. Second, in lymphoma, it's okay to kill all B cells because humans can survive without B cells with additional treatment. But non-B cell tumors don't have this same ability, so finding targets for CAR T cells remains a major challenge. The targets of CAR T cells, or anti-tumor immune responses in general, are generally classified into 2 categories: tumor-associated antigens and tumor-specific antigens. Tumor-associated antigens can serve as good targets for anti-tumor immune responses because they're expressed across tumors of different individuals and in all cells of a particular tumor type. CD19, for example, is a tumor-associated antigen which serves as the target for CAR T cell therapy in lymphoma, and it's an effective target to eliminate all cancerous cells. However, because the TAAs are also expressed in healthy tissue, when you use CAR T cell therapy against CD19, all B cells are killed and not just malignant B cells. Tumor-specific antigens, on the other hand, are more specific to cancer cells. These come from mutated proteins, and since the mutations are specific to cancer cells, they are not found in corresponding healthy cells. However, because they are mutated, their expression can vary widely between individuals' tumors or even between different cells of the same tumor. This makes them a really challenging target for immunotherapies because you need to identify a tumor-specific antigen which is widely expressed across different tumor types and is expressed in all cells of that particular tumor. Current CAR T cell therapies primarily target tumor-associated antigens in tumors, where it is viable to eliminate all cells of that lineage, both the healthy and the cancerous cells. But future CAR T cells are targeting tumor-specific antigens, which are normally derived from commonly found mutations across different tumor types. So we just discussed how tumor-specific antigens are often derived from mutations, and cancer is often thought of a disease of mutation, and most people think of genetic mutations first and foremost. But cancer cells also contain a lot of epigenetic mutations, with epigenetics being defined as any alteration to the chromatin, which is the folding structure of the DNA, rather than alterations to the DNA sequence itself. Epigenetic modulation occurs through enzymes like DNA and histone methyltransferases and acetyltransferases, which can add methyl and acetyl groups to the DNA or the histone proteins that the DNA is wrapped around to control how loosely or tightly the DNA is wrapped. And that, in turn, controls how accessible that portion of the DNA is for translation into RNA. Cancer cells can take advantage of this by using epigenetic mutations to control which parts of the genome are expressed or not. So they can either silence a tumor suppressor gene or activate an oncogene. And these are frequently mutations in the epigenetic enzymes and the histone proteins that the DNA is wrapped around. There are also epigenetic patterns that are found in cancer cells that differ from healthy cells, and patterns which are specific to individual tumor types, and these are currently being explored as markers for diagnosis and classification of various tumor types. Since cancer cells have epigenetic patterns which differentiate them from healthy cells, drugs that target these epigenetic enzymes can elicit cancer-specific effects. One of the effects of epigenetic treatment of cancer cells is known as viral mimicry. When you treat with an inhibitor of an epigenetic modulator, this can lead to transcription and activation of transposable elements within the genome. Elements like short interspersed nuclear elements, known as sign elements, can result in formation of double-stranded RNA in the cytoplasm when they are expressed. And these structures can activate innate immune sensors that can result in an interferon response. Interferon responses in the tumor microenvironment are known to be stimulatory, and they can activate previously inhibited immune responses. Because of this, we can use the beneficial properties of both of these therapies, epigenetic drugs and CAR T cells, to overcome the weaknesses that they each have individually. So you can use tumor-specific antigens targeted by the CAR T cells to gain specificity, and then use viral mimicry with an epigenetic drug to stimulate an immunostimulatory microenvironment within the tumor and allow endogenous T cells and immune responses to also be activated alongside the CAR T cell killing. So this may sound really great conceptually, but how close are we to actually using this idea in reality? Well, there are current clinical trials in glioblastoma using a mutated form of a growth factor receptor as a target for CAR T cells. This mutated form of EGFR is commonly expressed in glioblastoma tumors, but not in any healthy cells, making it a perfect tumor-specific antigen target of CAR T cell therapy. This clinical trial showed really exciting results demonstrating that this tumor-specific CAR T therapy increased patient lifespan. However, it was also found to recruit regulatory T cells, an immunoregulatory population which can dampen the anti-tumor immune response. So how can we address this immunosuppressive tumor microenvironment? Studies in mice and humans have validated that epigenetic drug treatments in tumors activates transcription of transposable elements, which then stimulates an innate immune response in the tumor microenvironment. Data from patients also tells us that expression of transposable elements in the tumors is correlated with increased immune infiltration, making the tumor cells more visible to the immune system. Finally, many studies have correlated increased immune infiltration with increased response to immunotherapies and better overall survival. This demonstrates that epigenetic therapy could serve as the perfect counterpart to CAR T cell therapy directed against tumor-specific antigens, combining the tumor specificity and killing ability of CAR T cells with the immune stimulation of epigenetic viral mimicry. Overall, finding tumor-specific targets and the immunosuppressive microenvironment remain major challenges in developing future cancer immunotherapies. But using tumor-specific antigens like mutated EGFR as targets of CAR T cell therapy in combination with epigenetic therapy to induce a viral mimicry interferon response could serve as a potential combination therapy that overcomes some of these challenges, and one that I'm really excited about personally. I hope you're as excited as I am about the future potential of cancer immunotherapy, and thank you so much for taking the time to listen.